Good evening. I am Ziad Buzarfawi, reporting live from the Computational Medicine Lab at New York University under the partnership and the affiliation of NYU Tandem School of Engineering, NYU Langone Health, and Tech for Health Institute. Tonight, we're tuning into the stomach's hidden rhythm to tackle long COVID mystery that still leaves millions feeling sick. Depending on the study that you read, between 3 and 79% of long COVID patients keep experiencing nausea, bloating, or pain, strong hints that the nerves controlling digestion have lost their beat. Inside the stomach, there are actually 5 anatomical regions that you can take a look at it like in the figure. But the most important thing are the tiny pacemaker cells, interstitial cells of Cajal, ICCs, that keep the smooth muscle cells marching at 3 cycles per minute. When that timing slips, digestion stalls. My summer project asked a direct question: can we detect the lost rhythm early without scopes or surgery using surface electrogastrography under the name of EGG? Instead of recruiting new volunteers, I downloaded a published classic motility dataset from this research paper of 19 that consisted of 19 healthy adults, 7 women, 12 men, average age 25, no digestive disorders, and no medications. Each participant wore 3 small electrodes over the stomach wall. We have 20 minutes recordings per person, one while fasting, one after a meal, worth 304 calories, which consisted of oatmeal plus orange juice. Record this run at 2 samples per second, giving 7,200 samples per channel. The step 1, which is the filtering, but at first there's a raw EGG signal. So the raw stomach signals pick up breathing, speech, and everything. They are really noisy. So the step that we have to do is cleaning, which is filtering. So a third older Butterworth bandpass filter kept only the 0.03 to 0.25 Hz window where real gastric waves live. Picture noise cancelling headphones for your gut. Step 2, we have to find the tempo using the frequency dominant lattice. What we had to do, a fast Fourier transform, FFT, which turned the signal into a power spectrum. The tallest peak tells us the stomach's dominant tempo, normally around 3 cycles per minute, but was within the interval of 2.7 up to 3.3 cycles per minute. So step 3, the peak detection. Guided by that dominant frequency, which is the tempo, I've written a MATLAB script that actually tags every true slow wave peak and logs its timing, which is in here, green triangles. And then we have to detect the non-slow wave peaks. Anything that doesn't fit into this category becomes a non-slow wave peak. The category of slow wave peaks means doesn't fit that category, non-slow wave peak, which is in here depicted like in blue circles. Key results. After the meal, the average slow wave frequency rose from 2.76 to 2.98 cycles per minute, an 8% jump. Slow wave peaks increased by about 4 per minute. Non-slow wave peaks hardly moved. All changes in the slow wave group were statistically significant. Why it matters? Why does it matter? Because a stable non-slow wave peak line, non-slow wave line can act as a baseline. If long COVID scrambles and the stomach's wiring, those extra peaks should spike. Here comes the physiological model. Signal analysis tells us what changes, but not always why. That's where our new physiologically based mathematical model that we're still working on, that we have to simulate how those electrical waves drive stomach muscles and how nerves modulate that process. The model actually links each EGG pattern to specific issues like delayed emptying or autonomic nerve fatigue. Imagine future patients receiving a personalized report that says rhythms such as mild gastrointestinal autonomic dysfunction. Here's what is trending, what to do next. Significance and impact. Together, the data-driven analysis, the model paved the way for a quick sticker-based checkup that could replace invasive scopes, slash costs, and extend care to rural clinics. Advancing the United Nations Sustainable Development Goal 3, good health and well-being. There's a statement of that Sustainable Development Goal 3. But next, which is within the process, I have to process real long COVID patient data through this pipeline and the model that actually explained if the rhythms are offbeat, we'll have finally a clear non-invasive marker for gastrointestinal autonomic dysfunction and the roadmap for targeted treatment. From NYU's Computational Medicine Lab, I'm Ziad Buzarfawi. Thanks for watching and keep your health in rhythm.